And welcome to Grand Rounds. I have the pleasure of introducing Dr. Andrea Dunaif. She is a native New Yorker. She graduated first in her class from Columbia University College of Physicians and Surgeons. She then went on to complete her training in internal medicine at Presbyterian Hospital in New York City and her subspecialty training in endocrinology and metabolism at the Massachusetts General Hospital. Her first faculty appointment was at Mount Sinai, where she remained for 10 years, rising to the rank of associate professor. Dr. Dunaif went on to hold a number of leadership positions in academic medicine, including the inaugural director of women's health at Brigham and Women's Hospital and director of Harvard Medical School's Center of Excellence in Women's Health. At Northwestern University, she served as the Charles F. Kettering Professor of Endocrinology and Metabolism and Chief of the Division of Endocrinology and Metabolism and Molecular Medicine. She was director of the NIH-supported Northwestern University Specialized Center of Research on Sex Differences in the Northwestern University Building Interdisciplinary Careers in Women's Health K-12 Career Development Award. She returned to Mount Sinai in 2017 as System Chief of the Division of Endocrinology, Diabetes, and Bone Disease of the Mount Sinai Health System and the Lillian and Henry M. Stratton Professor of Molecular Medicine at the Icahn School of Medicine. Dr. Dunaif is internationally recognized for her research on the fundamental mechanisms linking metabolism and reproduction. She has literally created an entirely new field of endocrinology related to investigation of insulin resistance and its consequences in polycystic ovary syndrome, or PCOS, the most common hormonal disorder of reproductive aged women. She has shown that PCOS is a leading risk factor for type 2 diabetes mellitus and used modern genomic approaches to discover causal pathways. Most recently, she has used machine learning techniques to identify genetically distinct PCOS subtypes. She has had continuous NIH funding as a principal investigator for more than 3 decades and has published almost 200 scientific articles and book chapters that have more than 55,000 citations, 30% since 2019. Dr. Dunaif has received numerous awards and honors, including the Endocrine Society's highest award for patient-oriented research, the Clinical Investigator Award, as well as the Arnold Adolf Berthold Medal Prize from German Endocrine Society and the Ricardo Aziz Career Award from the AEP-COS Society. She is listed in Research.com's Best Researchers in the World in the discipline of medicine. Dr. Dunaif has been elected to the American Society for Clinical Investigation and the Association of American Physicians. She received a doctor honoris causa from the University of Athens Medical School. She is a past president of the Endocrine Society, a former associate editor of the Journal of Clinical Endocrinology and Metabolism and of Obesity, and a past chair of the NIH Integrative and Clinical Endocrinology and Reproductive Study Section, and a former member of the NIH Office of Research on Women's Health Advisory Committee. Please join me in warmly welcoming Dr. Dunaif for Grand Rounds.
Well, thank you very much, and it's really a pleasure to be here. I particularly enjoy talking to internal medicine audiences because I think this condition really is a mainstream internal medicine disorder, and you probably just don't recognize that you're seeing it. It's also a model for sort of how we can start to untangle all the kind of clinical syndromes that we have in medicine where we don't know the cause, we just describe them, and how we might be able to work to elucidate the causes. This is my disclosure. I've spoken for Quest. So here are the learning objectives: to recognize that PCOS, polycystic ovary syndrome, is a distinct type of genetic disorder. It's what we call a complex genetic disorder. It's non-Mendelian. We'll look at how we can understand the causes through genetic approaches, and then I'll talk about some data-driven approaches to classifying it. So, why should you even care about PCOS? Well, it's incredibly prevalent. It's 5 to 20% of women worldwide, depending on how you diagnose it. Very expensive for US healthcare, about $8 billion. It presents right at when menses start. It can often be diagnosed So we have the opportunity to intervene to prevent some of the really substantial long-term cardiometabolic consequences. And what are those? Major increased risks for type 2 diabetes at a significantly younger age of onset. We're talking about girls and adolescents and young women in their 20s. It's also a leading cause of obesity, and in fact, Novo is marketing to PCOS is one of the disorders that its GLP-1 agonists are beneficial for. Since it's genetic, male relatives have a phenotype, I'll show you. There may be increased risk for cardiovascular disease, and it's also what's now being recognized as an atypical form of type 2 diabetes, and these tend to have frequently a genetic basis and are particularly good to study to understand the genetics of diabetes. So here is some work that Rebecca did when she was working with me. And this is looking at healthcare utilization and to show you that internal medicine and family medicine see more of these women than OBGYNs. And cardiology, and here, these are women 18 to 40, they're seeing cardiologists. So they're major users of internal medicine healthcare resources. And again, this is sort of the spectrum. They're seen when they're young because of their irregular periods, sometimes excessive hair growth. In their 20s, they have fertility issues. They have adverse pregnancy outcomes such as hypertension in pregnancy, preeclampsia, which are now being recognized as major risk factors for later cardiovascular disease. Here in internal medicine, we're seeing them for obesity, metabolic syndrome, prediabetes, diabetes. They're seen in the liver disease program for their metabolic dysfunction-associated steatotic liver disease. They have sleep apnea, increased prevalence, cardiovascular, and also anxiety and depression is increased. So really, this was just considered a kind of benign reproductive disorder until the early 80s. And what set the stage for kind of relooking at PCOS was a description of genetic disorders of extreme insulin resistance, which were associated with features of androgen excess, hirsutism, and also acanthosis nigricans. And these turned out to be due to mutations in the insulin receptor. These are monogenic disorders. And then it was recognized that an entirely genetically distinct group of insulin resistance disorders, the lipodystrophies, which are due to a variety of genetic defects in fat development, also when they occur in premenopausal women are associated with high male hormone levels and ovulatory dysfunction. They look like PCOS. So several genetically distinct forms of insulin resistance with this common feature of androgen excess. And that's what intrigued me during my fellowship when I started to notice that my patients with PCOS looked a lot like these rare disorders. They had acanthosis, they had hirsutism. So really the focus of my career has been trying to understand why these are associated, and I'm going to tell you about the genetics that we've done recently. So PCOS is a classic syndrome, which comes from the Greek just meaning events or traits that you find together, and you can describe together, but the cause is unknown. So for PCOS, what we have is first disorders of the regulation of reproduction, the gonadotropins. So higher LH, lower FSH, because GnRH, gonadotropin-releasing hormone, pulse frequency is increased. There's increased in ovarian androgen production, and the adrenals can contribute. There are multiple follicles, which are what has the oocyte, and they're arrested in development. And a marker of that is anti-Mullerian hormone, and that turns out to be very interesting because recently it's been learned it also feeds back on the hypothalamus to increase GnRH. So anyway, we have increased testosterone, and then it was recognized in about 1980 that these women were also hyperinsulinemic and due to profound insulin resistance and frequently obese. But completely unknown what initiated this syndrome, like many other things like type 2 diabetes and obesity. So how do we diagnose it? There are diagnostic criteria you'll read about, Rotterdam and NIH, but they're totally based on expert opinion. There's no mechanistic studies that go to what we should use for diagnosis. So hyperandrogenism, HA, ovulatory dysfunction, that's just long, irregular menstrual cycles, and polycystic ovarian morphology. And we get what's called the NIH phenotype, which is the group who have the irregular menses and the hyperandrogenism. And I call this sort of internist PCOS because these are the people who are at high risk for metabolic abnormalities and very easy to detect if you just take a history of a woman's menstrual cycles. And then there are a couple of milder forms of it that it's debatable where they should be. They're not associated with insulin resistance. So what I'm going to talk about is some genetic approaches to identifying, understanding PCOS. I'm going to talk about GWAS, genome-wide association studies, Mendelian randomization, which I'm sure you're reading a lot about. There are many, many papers coming out. Rare genetic variation, and then looking for additional phenotypes in men, children, and then trying to find ways to understand this disorder and ask the question of, is it really multiple disorders, and then will this lead to new classification and the holy grail these days, precision medicine. So what's a complex genetic disorder? Well, unlike a Mendelian disorder where there's either dominant or recessive mode of inheritance, usually these genes are fairly penetrant, so you tend to get a phenotype. Complex disorder is multigenic, many susceptibility genes working together with environmental factors to give you a clinical phenotype. And so, you know, height is a classic polygenic trait. Here we have Willie Shoemaker at one end of the distribution of height and Wilt Chamberlain, but really many, many genes contributing. And the disorders that we see commonly in internal medicine that have a genetic basis are complex traits. Type 2 diabetes, cardiovascular disease, asthma, obesity, and PCOS. So with the sequencing of the human genome, it became possible to effectively map the entire genome in individuals at a very, very large scale. And this enabled being able to look for the genes that cause these complex disorders, because you need very large sample sizes of cases and controls. Each individual's DNA is run on a single array that has now more than 1 million single nucleotide variants tagging across the genome, and then you can see where there's genetic variation. These are called Manhattan plots because this looks like the Manhattan skyline, and you have a level of significance which you have to correct since you're doing like 1 million or more tests, so 5 times 10 to the minus 8, and you find signals on chromosomes that are associated with your disease of interest. And these studies have turned out to be incredibly enlightening for understanding the pathways that cause disorders like PCOS. And type 2 diabetes is a really good example because before GWAS, everyone thought that insulin resistance caused type 2 diabetes. With GWAS, the signals that came up were mainly in beta cell genes, either number or size. And now the concept of type 2 diabetes is that you have to have beta cell dysfunction as sort of your final step to decompensation and glucose tolerance to get type 2 diabetes. and that this is more important than insulin resistance. So now we've done a number of large GWAS in PCOS, and both in European populations and in Han Chinese. And the pathways, which is consistent with the pathophysiology that the genes are implicating, are neuroendocrine pathways. We're getting genes that regulate gonadotropin secretion and action. Not surprisingly, reproductive genes. This gene I'll show you a little bit more about was discovered in GWAS, turns out to be a very important regulator of androgen production in the ovary. They're genes that regulate other functions in the ovary. And then there are DNA repair genes, which are associated with age of menopause. And in fact, women with PCOS go through menopause at a later age. So this is sort of an anti-aging variant in the ovary, which I think would be fascinating to see other tissues, what the aging process is in these women. And then finally, metabolic genes have come up. FTO, which is a major susceptibility gene for obesity, and then genes for insulin resistance, sex hormone binding globulin. So really, reproductive, neuroendocrine, and metabolic pathways appear to be the major genetic pathways leading to PCOS. And this is an example of how GWAS can lead to the discovery of new biology, new pathways. This signal at DEN-D1A came up in the first PCOS GWAS, and it was not known to be associated with PCOS or ovarian function. It's an intracellular trafficking protein, but it turned out to be a very important regulator of testosterone production and overexpressed in the ovaries of women with PCOS and the theca cells, which make androgens. But the exact genetic variant that causes this, and this is very, very common in genome-wide association studies, that mapping the precise genetic variant that's causal is extremely difficult. The way we're finding these genes is really just through marker variants. So another thing that GWAS has allowed us to do in PCOS, as I mentioned when I was telling you about the diagnostic criteria, they're based on expert opinion. People argue over them, which ones to use. And there was a large enough sample size in a meta-analysis that we did several years ago to actually ask the question. And you could see doing this in any disorder where you had enough sample sizes to contrast different criteria for different things. So we took NIH criteria, non-NIH, Rotterdam, those additional 2 types. And this was an interesting study because we also had a very large population of self-reported PCOS from 23andMe. And essentially, they're all genetically similar. So the diagnostic criteria, it's now been replicated, don't identify genetically distinct forms of the disorder, so at least we shouldn't be fighting about that. And you can even drill down on this more. And here what we did, these are just the lead SNPs in the meta-analysis, and looked, obviously they're all associated with PCOS, so we wanted to see which ones were associated with 2 of the features of the diagnostic criteria, irregular menses or polycystic ovarian morphology. And you can see that there's almost complete overlap. So one of the arguments is, do we have to do ovarian ultrasounds in these patients? And it's a big disincentive, I think, for other specialties caring for them. No, you can take a menstrual history and you're getting the same. Another approach that's been enabled by having all these GWAS data and so having many, many traits that have had genome-wide association studies, and we can find genetic variants that predict the trait, you can actually ask, do sort of a form of a randomized clinical trial, except you're using genetic variation, and you're asking the genes should be randomly distributed, And so if you have genetic variants that are a good proxy for the exposure that you're interested in, let's say body mass index, you're able to ask if those genetic variants are associated with the outcome you're interested in, and you're not going to get the same confounding by environment. So there are limitations to this, but you're actually able to ask without additional environmental confounders if there's a causal relationship between genetically determined BMI and PCOS. And in fact, you can show, at least in this analysis, that genetically determined BMI does contribute to PCOS, is causal, but the other way around, there's no association. So there is a contribution of higher BMI to the development of PCOS. Another hypothesis in the field has been our male hormones, testosterone, kind of a final common path for the disorder. So do you have genetic variation that gives you high testosterone levels, and then those, either through even gestation, but certainly early development, do they then imprint or program and cause other features of the syndrome? And this hypothesis came forward. We found a long time ago that if you look at families, the consistent phenotype that you see is high androgen levels, and in fact, you even see it in the men. You can make an animal model of PCOS that's very realistic, very similar to the syndrome, by giving androgens in utero to the mothers and in high doses, so they cross the placenta, and so you can create PCOS in the offspring. I told you about DEND1A, this androgen regulating gene being very important. So with Mendelian randomization, using genetic variants that are highly correlated, predictive of testosterone levels, you can show that testosterone does indeed play a causal role in the development of PCOS. And this is a study from the UK Biobank, and also able to show that it contributes to the development of type 2 diabetes, but just in women, not in men. So this has really been powerful for sort of starting to dissect the main pathways that contribute to disease and what the direction of the relationship is. Another approach that you can use is to look at sharing of genes. So we now understand that most diseases are just highly, highly polygenic, and that a huge number of genes contribute to the development of disease, and what matters is what tissues they're expressed in and how they're regulated, and that there's a lot of overlap. And you can actually measure this overlap between separate disorders. And so genes that contribute to fasting insulin levels, highly correlated, highly overlapping with PCOS genes. Obesity genes, type 2 I just showed you, and depression genes, coronary artery disease. So we can start to see another reason, in addition to just causal relationships, that disorders may have features of several disorders, this sharing of the same genes. And then finally, it's proposed that in addition to all these peripheral genes with small effects, there are many fewer central genes that have direct effects on the phenotype, and they're called core genes. And we can sort of get an idea about them by looking for rare genetic variation. And what we find in PCOS is indeed there are some genes that are not surprising, AMH, and again, it's receptor and DEND1A, and these all have rare genetic variants. And so these are probably core pathways that are impacted by these other peripheral pathways with smaller effects. Now, what about the other phenotypes? So is there a phenotype in men? And one way you can do this is you can use polygenic risk scores. You just sort of, it's a summation of all the genes that are associated with the disease, done, adjusted in several ways, and ask if those polygenic risk scores are associated with other phenotypes and outcomes. And so here we're looking at polygenic risk scores increasing, body mass index, and this is in men. And so you can see that higher polygenic risk for PCOS is associated with higher BMI in men. And it's also associated with coronary artery disease, but the mechanism of the association is through higher BMI. But observational studies done a long time before the genetic studies also showed if you looked at the first-degree relatives, they had more metabolic syndrome and more obesity. So kind of now I want to change the focus to start to ask the question, is PCOS one disorder or multiple disorders? And so one way of thinking, and you can take your favorite disease and probably put together a figure like this where you decide one thing causes everything else. So this is just a figure showing how insulin resistance can cause all the other features of PCOS, and that insulin resistance and high insulin levels are the cause. So this sort of unitary etiology. Or another way of thinking, and this is the work of Victor McCusick, is that you can have a phenotype, and here this is short stature, this is the Little People's Convention, and all these people have genetic short stature, but the genes that cause it, they're multiple different genes. So you can get to a final outcome from many, many different pathways. And so how can you start to sort this out? And what we have now is access to huge amounts of biomedical data. And as a lot of initiatives here at Mount Sinai, the analysis of these data and looking for patterns in these data is a way to start to kind of, in an agnostic way, that is without a prior assumption, ask, are there subsets that are related to each other? And you can do multiple mathematical approaches. I'll show you just with clustering algorithms, but you can start to subset disorders into discrete categories. That's been very powerfully done in oncology, and it's coming to a disease near you. So you can do clustering, which is just asking mathematically what parts of data, of a set of data or traits or whatever, are more related to each other. And there are a couple of ways of asking this, but what you're trying to do is have the items within the cluster be closely related, and that between the clusters to increase the distance. So with PCOS, we now have done this in a number of large cohorts, but we just took cases with a very rigorously defined PCOS, the NIH, and they're from the US, the Netherlands, Greece, Korea, and going in, the only thing that's different between them is body mass index. Otherwise, all their hormone levels are similar. And we found that in all of these groups, we could identify discrete clusters. A cluster characterized more by reproductive traits, higher LH, higher sex hormone binding globulin, a cluster defined more by metabolic traits. Now you can say that's not surprising. Metabolic traits sort of run together. If you have high BMI, you have high insulin and glucose. But these clusters meet additional criteria, and I'll show you some supporting data. And these clusters are a feature of PCOS across populations, even in a very distant ancestrally population, East Asians, we're still seeing these distinct clusters, and we replicate them in, multiple independent cohorts. And here we're just looking at how the hormonal traits look. And, if you're in the metabolic cluster, you have high BMI and insulin. If you're in the reproductive cluster, you have high LH and sex hormone binding globulin. And what's interesting is the testosterone is the same. So that's not a differentiating. So that's a common feature. And then we have these distinct features. Also, if we can make our groups more homogeneous, we'll have better power when we do genome-wide association studies because we'll be removing noise. So here is a study that's combining populations, PCOS, European and East Asian, and looking for genetic signals associated with these clusters. And we find in the metabolic cluster, there's a very strong signal across genome-wide significant signal, and this is transethnically at this gene, C9orf3, which may be implicated in fat metabolism. And then in this cluster, or these are really the people who don't cluster, when we remove these groups, we get a very strong signal at this gene FSH beta, which is a gonadotropin, and regulates production of estrogen. And so we can show that these subtypes really are associated with distinct genes, suggesting that the clustering really is identifying biologically relevant groups. And we can take gene signals from GWAS and ask, are they associated with one of these subtypes? And so it's a way of perhaps better understanding which genes contribute to which subtypes. And we can see that this C9orf is a strong signal in the metabolic, but not so strong in reproductive or background, that the testosterone gene, DEND1A, across all subtypes, consistent with the trait data, and then this gene, theta, which is a diabetes gene, interestingly here is in the reproductive group, this gene, and these genes in the reproductive. So it's a way of starting to sort which genes are associated with which kind of outcomes. And this has now been confirmed by a couple of different approaches, other groups showing that there are these reproducible metabolic and reproductive subtypes. And you can even show that as girls with PCOS and adolescents have evidence for these subtypes. So this seems to be a pretty robust finding that there are at least 2 to 3 main groups of PCOS, which are genetically distinct. It's not one disorder. And hopefully that can lead to precision medicine approaches to target specific therapies based on subtype. We certainly need to do outcome studies. And here we're showing that there are additional features that if you subtype these women, you'll find the reproductive has higher AMH and oocyte counts, and the metabolic has lipid abnormalities. And this study is showing that IVF outcomes differ by cluster. So some efforts towards precision medicine. So the strength of these kind of approaches is that we're identifying these subtypes, and they're present across diverse groups. We can do gene discovery with our clusters, and they may predict outcomes. And the limitations of this approach, very sensitive to outliers. You really reduce your sample sizes, and so you really need large cohorts to do this sort of research. And this is an approach that's now being applied to a number of complex disorders, in particular type 2 diabetes. NIH had requests for applications to do this as a nationwide networked subtype diabetes using electronic health records. The work that's already been done is that you can subset diabetes into about 6 different subtypes, and it's different combinations of insulin resistance, insulin secretion, and obesity, and that these subtypes are associated with different combinations of known genes. They haven't been used for gene discovery yet. And you can get towards mechanistic insights by looking at which groups of variants are associated with your different diabetes subtypes. So what we can say about PCOS is that it's a complex genetic trait, that our GWAS variants implicate neuroendocrine, reproductive, and metabolic pathways in its pathogenesis, that the genetic architecture of the phenotypes or the groups defined by the current diagnostic criteria are genetically similar. They aren't behaving as distinct biologic entities. That there's rare genetic variation that's suggesting that important core pathways are androgen biosynthesis and AMH signaling. So we can start to put together kind of a holistic view of what's going on with our subtypes, these having more neuroendocrine features, metabolic, and the high androgen levels being something that's a feature across, that the common genetic variants, at least when we just look by the current diagnostic criteria, are similar, but the rare genetic variants are starting to sort out to implicate distinct pathways, and that there's an interaction with environmental factors. There may be some intrauterine exposures like testosterone, and also in adulthood, obesity may contribute. And that these approaches are really something I think we're going to see a lot of to try to do more precision medicine approaches to the common diseases. And that you can also use the hard clustering for gene discovery, and that these are going to provide data-driven approaches for diagnosis and classification of disorders. So just sort of to summarize, that what we currently are looking at is just similar. We're not capturing the heterogeneity with our current diagnostic criteria, and that moving forward and using these approaches, that it's very promising that we are starting to come up with discrete entities with different pathogenesis. And so I'm going to close here, acknowledge collaborators. These efforts are always very large because of the number of subjects you really need to do these analyses. And I want to recognize the group here at Sinai, and in particular Girish Nadkarni, who's been a big help in our clustering approaches. So with that, I would be glad to answer any questions.